Evening and to talk to you about annelids. You'll be able to admit people?
Yes.
Okay, great, terrific. Yeah, so I kind of fell into working with annelids. I have a longstanding interest in microscopic organisms and microscopic life. And it's my father was interested in astronomy, and I just kind of went the other way, just exploring the world when you are equipped with a little bit of technology to help you see things that are smaller. So that's how I fell in love with annelids, but I'm really broadly interested in organismal diversity. So I will share with you some things about annelids, and at the end tell you a bit about some of my research program. Okay, so what we'll do today is I will, I try to convey in my slides and what I'll share with you today, just some of the reasons why I think they're just such a fascinating group. So I'll give you an overview of the annelids, tell you a little bit about their diversity, their morphology, their natural history. I'll highlight some of the reasons why they're ecologically so important. Knowing that we are all here, people interested in the natural world, thought maybe I could tempt you into going out and collecting some aquatic annelids. So I'll tell you a little bit about how that can be done, at least the way we tend to do it. Give you an overview of some of the more common aquatic annelids that we have in Maryland. And then, like I said, at the end, I'll give you a couple of snippets about the kind of work that I do here at the University of Maryland. Okay, so first an overview about annelids. So I'll start off by saying that if you go to the Chesapeake Bay, or if you go to a creek, or if you go to a stream or a pond in Maryland, you will find annelids. It's not like, oh, will I find some? You will find some. They're really, really common in pretty much any kind of, relatively permanent body of water. And they are beautifully diverse. So if we look at think about annelids at the level of the phylum, so the broadest categorization that we have within the kingdom of animals, we have these phyla. And so the phylum Annelida, the segmented worms, includes a really broad diversity of worms. So they include things like bristleworms and mudworms and earthworms and leeches, but many, many others, giving you just a sampling of what their bodies can look like here on this slide. And you may be surprised to realize just how big this phylum is. It is about 20,000 described species, and that is an ongoing process. There's continuing descriptions of annelids, so it's a really big phylum. About 3/4 of them are aquatic. So although we on land here, most, like the first contact that most people have with annelids is when we're little and we're digging in the dirt and we find some earthworms. So there are thousands of species of earthworms on the planet, but most of the annelids are actually in the aquatic realm, so freshwater or marine. And I like to kind of put things into perspective. So, humor me for a minute here. I teach animal diversity, so I'm always bringing it back to, what do we have on the planet? So when we think about what animals are on the planet, we have about 32 animal phyla. So phyla meaning these major kind of body organizations, so major groups. And there are about 32 of them, and annelids are one of those, but they're really what we would call like one of the big 9. So ones that most people are familiar with. On the left here, the chordates, right? Our group, the bugs, insects, the arthropods, the mollusks, and then third here is the annelids. Also have things like cnidarians, jellyfish, and corals, and sea anemones, sea stars, and sea urchins are the echinoderms, nematodes, the roundworms, flatworms, the platyhelminths, sponges. And then probably you look at the bottom here and yep, all of those are additional phyla, but they're much less common. Some of them are very small and often considered kind of minor. But just to kind of zoom back out to make the point that annelids are one of our core major animal groups on the planet. I loved seeing this paper when it came out a couple of years ago, a first attempt to understand where is the biomass on the planet? Annelids actually show up on that. They are not insignificant at all. It's estimated by this first really remarkable effort to try to understand where the biomass is on the planet. When we zoom into the annelid component, the animal component, annelids, shown here in the upper right in this turquoise color, represent about 10% of global animal biomass. So this is really significant. The other big chunks are the vertebrates here, arthropods, of course, mollusks, so snails, slugs, and cnidarians, so the jellyfish and anemones. But annelids, really a sizable fraction, at least based on this first really major effort to estimate where animal biomass is. So let me tell you a little bit about the features of annelids, so that you can start getting a sense of what this group is like. They tend to have an elongated body shape, usually, except for the ones that don't. They tend to have segments that evolved at the base of that group, but usually there's a couple that have lost their segments. And so here's a couple of examples here, just representing that elongated body shape, those segments along the body axis, so that each unit has, often many parts of their organ systems are represented in each of those segments. But like I said, that's usually what they are, elongated and segmented animals. But evolution doesn't make things easy for us. And sometimes things that are, evolved early on and are present within much of the group just get lost in the lineage. So we have kind of disc-shaped annelids, and we even have annelids that have lost their segments entirely. They're called sipunculid worms. They were actually described as a separate phylum because people didn't recognize that they were annelids, but now it's very clear that they just lost their segments. But mostly elongated body with segments. They have a through gut, so the food comes in one way and out the other, which is not the case for some other groups of animals. They have an anterior end with a small brain near the end here. So on this picture here, can you see my cursor if I do this? Yeah. So that's the anterior end of this particular worm, and up here is a small brain. But they also have like a small brain, in a sense, in each body segment. So if you look along the length of the body, there's a little piece of the nervous system, and actually the central nervous system within each of those segments. So they have a distributed nervous system. We have most of our nervous system is in our brain, the central nervous system. They have it some in the brain, but also a little bit in each segment, which means that if they lose a body part, they can still do a lot, because there's still that processing center within the segments. And then they have some really cool sensory organs, just showing you a couple here, these kind of sensory extensions here on this beautiful psyllid, these very cool expanded eyes in this kind of pelagic open ocean polychaete. They are worms. What is a worm? A worm is just something that's long, skinny, and doesn't have limbs, right? There's a lot of different kinds of worms. This is just one kind. So we don't see limbs within annelids, but we do see all sorts of interesting appendages. So some of them have these lateral appendages that are called parapodia, meaning beside foot, meaning things that are kind of like a limb but are not. So you'll see that here, for example, in this middle one that has these very long extensions of the body that are called parapodia. This one actually swims very well. You see them a little smaller on this one. Some of them have tentacles at the anterior end. So we've got kind of like these long spikes here, but here's one that has tentacles, and sometimes other body extension. Oh, so up here also beautiful tentacles on this sabellid polychaete here. And then there's other things that, evolve in certain lineages. So here's one that has all these projections along the body axis that are thought to be used for respiration. So it's a high surface area structure like our lungs, except that it's sitting out in the water. And then here's one that has kind of tentacles, but at the tail end, because they actually breathe partly they get a lot of their oxygen through their hind end because their head is often deep in anoxic, nasty stuff where they're feeding. And so their accessory respiratory organs are actually at the tail end. So things sticking out of these animals. They also, many of them have bristles that are called chetae. If you know something about arthropod morphology, you might have heard the term setae. It's the same, it's just the same kind of root. In annelid biology, we tend to use the word chetae. And those are just the hard bristles that stick out of the body, usually within each segment. Here you're seeing some of them along here. Here's a high magnification view by SEM showing those hard, almost like little fingernails or spikes sticking out of their body. And here you're seeing those bristles sticking out this way too. And they use those for all sorts of things. One of them for, just having good contact with their environment so that they can push themselves around. Sometimes they're used for picking up sensory information like vibration. And they can take a lot of different forms. So I'm showing you here just some of the morphology of those chetae. And even things that don't look bristly, like an earthworm, if you look at high magnification, you will see those chetae sticking out, just tiny little bristles sticking out that help them kind of combat, just be able to increase that friction so that they can push themselves through the environment. And of course, I say mostly, usually they have chetae because, of course, there's some groups that have also lost the chetae. Back to our Simpunculid, the one that lost the segments, it also got rid of all its chetae. And leeches. Leeches are a group of annelids, but they have lost their chetae as well. They're just, moving differently. And so that's been lost. They're mostly soft-bodied, but some of them make tubes. So here is one that's just soft-bodied. The only hard parts on this animal here, and this is pretty typical, is those chetae that are sticking out. But some annelids, and this has happened many times independently, have evolved to be able to live in tubes that they make themselves. So the deep sea tube worms, they live in these calcareous tubes that they secrete. This is another group that also secretes these calcareous tubes. Some of them, called parchment worms, make almost like a papery tube that's quite pliable, but that's secreted. And then others that will make kind of a gluey substance and then just pick up foreign debris and live in that. So a lot of ways for these animals to protect their soft bodies within these outer coverings that they make that are not formally part of their bodies, but they're often made by the animal. So tube dwelling has evolved a number of times within this group. All right, and then most of them are aquatic. Like I said, the majority of them are marine, but there are brackish water and freshwater annelids, quite a lot of them actually. And back to the familiar earthworms, there's a good number that are terrestrial, primarily in moist habitats. You won't find them places that are really arid. They are called benthic, meaning that they associate with, primarily they associate with surfaces or substrates. So we typically find annelids within sediments, whether they're terrestrial sediments or marine or freshwater sediments, or on those surfaces. So if there's something submerged in the water, you might find annelids on top of rocks or shells, as well as deep within the soils or the sediments. And I say mostly because, actually, I'll just skip this one because these videos weren't playing well, but there are some that are entirely what's called pelagic, meaning they only live in the water and they don't touch the bottom. So there's, I encourage you to Google for a video of Tomopteris, which is a beautiful annelid that lives its entire life in the water column and swims using these big parapodia that they use to basically paddle around in the water. And reproduction, also really interesting and diverse within this group. We've got here on the left, cute little larvae. So a lot of the marine annelids have a form, a juvenile form that looks quite different than the adult. And so here is one of these little larvae with cute little eye spots and a little gut here that's filled with some little oil droplets that it's going to be able to use as food while it's still growing and before it takes up too much from the environment. So many of them have an adult form, but then a separate larval form. Another cool thing that we see in terms of reproduction within this group is what's called hermaphroditism, where animals have both male and female parts, and they're making both eggs and sperm. And so that's evolved a number of times across animals. And one big origin is within the annelids and the group actually that led to earthworms and relatives, which is why if you've ever seen earthworms kind of paired up like this, they're mutually exchanging sperm, and both of them will have sperm that they can use to fertilize the eggs that they have. So an interesting situation where you have both male and female reproductive structures within the same individual. Another cool example of annelid reproduction are these little psyllids. So it's a little marine annelid that it makes its eggs within its body, and then when those are fertilized and they are laid, they lay them essentially on their stomachs, and they carry around, like good mamas, carry around this big packet of eggs until they're ready to swim off and be little juveniles swimming around. So you see these very cute little marine annelids that are carrying around a big packet of their eggs. And if you've heard of pololo worms, another very cool type of annelid. These are annelids, and this has happened a number of times within the annelids, that reproductive forms, right? They're mostly living on the benthos, as it's called, like on the bottom of the of the ocean or of the stream or whatever. But in these marine animals, sometimes certain lineages, when they become reproductive, they actually become a swimming form, and they all swarm up towards the surface of the water. And there, that ascent is synchronized so that you just have tons of these worms from certain species coming up into the water column and essentially exploding so that the eggs and sperm are released into the water. But because the animals have swum up to a surface, it's fairly concentrated, and so fertilization can take place. So you get these beautiful swarms. You see this I've never seen it, I would love to sometime, but in Indonesia, in that area, New Zealand, parts of Australia. And if you're, feeling hungry on a Thursday night, it might tempt you to consider eating some pololo worms. They are considered a delicacy. This happens just once a year, but they have like these huge swarms. And so native cultures have harvested those for food. And then a type of reproduction that I particularly love that we see within the annelids that's evolved several independent times is what's called, it's a type of asexual reproduction where the animals make worm chains. So let me kind of walk you through this one. Here's the head of a worm. Here's the tail of a worm. And in the middle here, you see a second head. So it goes head, tail, head, tail. And here in this beautiful marine version, there's the head, that's its tail, but then that's the tail and the head of one that it's going to be kind of budding off. And here's another one. It's like a conveyor belt of little individuals that it makes that are being kind of generated from here. So you get these beautiful worm chains. So a very cool phenomenon that's evolved repeatedly within annelids. So certainly most of them don't reproduce this way, but it's happened a number of times. And then another really cool thing about annelids is that some of them are very, very good regenerators. So here is a little marine one that's reproducing, that's making a new head. So where you see that lighter tissue, that's the new head that's being built on top of this older body. And then here a group that we work on, these are these little freshwater ones. If you take off the head or the tail, they can make a new head, shown here, and a new tail, shown here, within like 2 to 5 days, very, very fast. And that involves making a new head, a new mouth, a new kind of throat region, a new brain and those nervous system ganglia. So really extensive regeneration in these animals. Just a close up here to show you the new mouth. So the amputation plane, or where it was cut, was around here. Here's that new foregut. That's the brain right there. It's making a new little snout. There's the mouth. And this is probably within about 4 days of being cut. So all sorts of cool things. In terms of feeding, most annelids feed on things like sediments, algae, suspended particles, small bits of things, but feeding ecology is really diverse. We have, for example, in those deep sea vent worms, their guts are no longer serving a purpose, involved with like taking in food from outside and digesting it, like most animals do, but their guts are filled with symbiotic bacteria that can basically make food from the surrounding kind of chemical composition of the water. And so these animals no longer ingest food, they just harvest their bag of bacteria that they hold in their guts. Some other cool and unusual modes of feeding within annelids. Here's one of these predatory marine worms that will sit mostly submerged within the sediment and wait for something big like a fish and snap it with these big kind of hooked mouth parts, and they will just drag it right into the sediment and devour it. So some that you wouldn't want to get too close to. Leeches are in this group, and so here's a beautiful leech on a beautiful turtle, taking a blood meal from either a turtle or fish. Also have leeches that will feed on them. And here's another unusual feeder within the annelids. This is one that lives in the kidneys, in the urinary tract of these little frogs. So yeah, not the UTI you want. Not that anybody wants a UTI, but this one is unusual. You've got these little worms in there in the urinary tract of these worms. There's of these little frogs. So some cool feeders, but most of them are feeding on things like sediments, algae, and so just small particles. Okay, before I transition to the next part of the talk, I want to zoom back out and come back to the slide of the 32 major animal groups and say that there are a lot of different kinds of worms, and annelids are just one of them. So you can't just say, oh, I work on worms. It's like, yeah, but which kind? So about almost half of the animal phyla, meaning almost half of the major types of animals that we have today, which I've given you a representative picture for each one of those. 15 of them are wormy, meaning they're longer than they are wide. They don't really have limbs. They might have some structures that stick out, but nothing that you would call a limb. And so these are all wormy. And I've highlighted them in yellow here just to show you that, yes, we have annelid worms, but there are things called flatworms, and they have really different structures. There are things called roundworms or nematodes. They have really different structures. We have things like hemichordate worms called acorn worms, a bunch of different other ones that the names probably wouldn't mean anything to you, but just to highlight that there's that form has evolved many times, and annelids are just one of them. Okay, so a little bit about their ecological importance. They're a really important food source. Why? Because of some of the reasons that I was just highlighting, which is mostly they're unprotected. They are, tasty, long, fleshy morsels of food, and they are, if they can be, if they're found, many of them are quite defenseless. So some of them do definitely have some forms of protection. But that means that within our mud flats, within our freshwater, within our ocean sediments, and certainly within on land, we've got all these, soft, tasty morsels of food. And a lot of animals are adapted to feeding on small invertebrates, including annelids. And given that annelids are so abundant, they can be a really important food source for higher trophic levels. So here's a beautiful bird eating a little marine polychaete. And just to bring it back to this area, they're really important for young fish. So juvenile fish are, many of them consume large quantities of annelids. There's a study that just came out in 2023. Really glad to see this study out of the group that's at SERC, at the Smithsonian Environmental Research Center, that was looking at what are juvenile striped bass eating. And a picture that I'm not showing you here is if you just open up the fish gut and say, okay, what do I see in there? It's really hard to see what's in there. And as you can imagine, the things that are kind of have hard body parts, you'll be able to recognize, and things like annelids that are mostly soft body parts, it'll just look like mush. There's very, very little to go on. And so I think it's been underappreciated just how important annelids are, because often we can't see them if all we do is just open up the gut. But this study used a molecular approach so that they sequenced, they got the fish gut content and said, okay, we're going to see what kind of DNA sequences we find in here. So that we can tell what are these animals eating, even if we can't see it and recognize it based on the visible remains. And you'll see here that about 39% of what these juvenile striped bass are feeding on is crustaceans. But the next biggest group, for about 20% of the gut contents, are here worms. You now know there's a lot of different kinds of worms. Here they mean annelids. And so a very substantial fraction of Chesapeake Bay juvenile fish are probably feeding substantially on polychaete annelids. And then one more kind of broad ecological role that annelids play is in sediment processing. So I said many of them live in or on the sediments or in the dirt, and their activity burrowing through the dirt, ingesting dirt and having it pass through really churns up the sediments, and it changes how deep the water can go in if it's a submerged sediment, how deep the air can go in if it's in a terrestrial environment. It changes what bacteria are able to grow there and in what kind of numbers. It changes chemical properties. So they are sometimes called ecosystem engineers because they have such a profound impact on their world, on their local habitat, so that then other organisms either can or can't persist there, or the other organisms in that community are impacted by the presence of, in this case, annelids, because they change the habitat so significantly. Okay, so hopefully I've started, piquing your interest, and you're probably like, okay, yeah, let me go try to find some. So let me just tell you a little bit, kind of, collecting annelids 101, and I'll highlight that at this point I'm going to zoom into the aquatic annelids. At some point you should have if you haven't already, have an earthworm specialist come out. That's a really different kind of community and biome. So I don't. I'm not your expert there, but I've spent a lot of time collecting aquatic annelids. So I'll show you how we do it. So first of all, like I said at the beginning, most water bodies have abundant annelid communities. So, that's one of the nice things of working on annelids is pretty much anywhere I go, if I want to collect some annelids, I'm going to find some. So this is a Paint Branch here, right just off of, right near in College Park. This is again SERC, so on the Chesapeake Bay, Smithsonian Environmental Research Center, two places that we certainly do regular collections from, and annelids are super abundant. So to collect aquatic annelids, you're often going to have to collect the sediment itself, right? Because many of them are physically embedded within the substrate, whether that's mud or sand, or even if they're living on things, you're going to probably need to collect that because many of them are on the small side. So you're going to collect sediment, you're going to collect aquatic vegetation. If it's in more like a pond, you're going to maybe scrape things like my student here is doing, scrape things off the dock, get some of that biofilm and all that material that grows on surfaces that are submerged, and you're going to collect that and bring it back. So we, take jars, and sometimes it's a peanut jar, and then somebody's like, anything goes, right? Just put something in a jar and then bring it back. It's hard to do all that stuff in the field. We always bring it back to the lab. And when it's hot, here are my two students, we put things in a cooler just so that things don't get too hot. It's also a good idea when you do collections to take a little, take a couple of jars or take a jar or two of water from that area, especially if it's a brackish area, so that you have some water to work with when you come back to the lab. And it's water that the animals are kind of adapted to. In terms of sorting for aquatic annelids, you're going to have got that sediment, that mud, that sand, that debris. And if you're looking for large annelids, you can put that in a sorting pan and just, put some water, shake it around. Annelids don't stop moving pretty much. So what you're looking for is annelids that are just, that motion. You're going to look for something that's kind of elongated, that looks like it has those segments, and that's just kind of moving around. They're usually, you pick them out based on that movement primarily. So if it's a large worm, you can put it in a pan or in a big glass bowl and just look at it, put some in there, put some water in there, look for that movement, pull some out with a pipette into a smaller dish. When you want to look for the smaller world, look into the smaller world, which is a lot of what we do. We use a swirl and decant method where we put a little bit of that mud or that sand or that debris in a flask. And we use an Erlenmeyer, but you can really use anything where you can swirl without getting yourself, completely drenched. Swirl so that the animals are usually lighter than the sediment is. So what happens is that the animals are suspended in the water by your gentle swirling, and then you pour, so you decant into either a dish, or if you're going to do a lot of that, into some kind of sieve that will let most of the water pass through, or the water pass through, but it'll trap animals that are at a certain size. So this would be a fairly coarse one. If we were using this one, we often use quite small ones because some of the smaller animals are the ones that we're often looking at. So for large worms, you can use just a pan. If you're interested in the smaller worms, you're going to probably want some kind of swirling and some kind of sieve. But although you can buy these and you can make some with certain very, very fine mesh sizes, at least for you might miss the really small ones, but you can even just use like a strainer. That works. And it'll at least keep some of the bigger worms. And then you're looking for body movements. And to give you a sense of the body's size, the smallest ones are about 3 millimeters. So really small. And the biggest ones, that are most common around here would be about 10 centimeters. So that's the range of things. Many of them are more on the scale of a centimeter or two. And then, if you have access to a microscope, this is where the beauty of the animals becomes apparent, and it's just so wonderful to do this. So here are my students, working with some of these specimens at our scopes. It doesn't have to be super high powered because many of them are still on the scale of, half a centimeter, a couple centimeter. So we often do some of our sorting actually under the scope so that we can pull out what we want, and we don't miss things that are small but still really interesting. But if but the exact power, is kind of, it'll just affect exactly what you see. But as long as you can, even if you don't have a microscope, you'll be able to see the larger animals. And if you want to see the smaller ones, then some kind of dissecting microscope is useful. In terms of identifying aquatic annelids, I'm putting here, I just pulled off some of the books off my shelf. So these might be older editions, but for freshwater, Thorpe and Kovich is great. Pennack is great. Pekarsky et al. Is great. These are, especially the top two here, have a fair bit. And it's not, technically too challenging. They're using characters that are, for the most part, ones that people can follow along with. There's also kind of more detailed keys that if you got really interested in a particular group, but these are great ones. I use these all the time. And in terms of the marine realm for that, just to have a guide, I like this one. It's got a whole bunch of different polychaete families, as well as some of the polychaetes are the marine ones, as well as the freshwater ones. So this is a nice guide for, I keyed something out just over the summer. I was like, oh my God, what is this worm? And it's like, oh, there it is. So these are nice resources. In terms of what features you might be looking at when you do this, it'll be focused on the morphology of the segments, whether they have some of these appendages, what the head and tail look like. Sometimes to get down to, to species, you might need to look under higher power at the shape of those bristles, those chaetae. And then for some of them, for example, earthworms and some other freshwater worms, you need to go into the internal anatomy. That's a whole other level of expertise that you need for that. But you can do so much without getting to that point. And all of these, I'm pretty sure all of these is just all external anatomy. So very accessible. Okay, so I'll highlight a little bit about aquatic annelids in Maryland, what you might find here. So first focusing on freshwater. Going from kind of biggest to smallest, one of the fairly common worms that you'll find around here is lumbriculates. That's the general name for them. That's the family name. They've got this beautiful striped head. They're very, very active movers. And this is one of the absolutely top regenerators of the animal kingdom. You can pretty much chop this up into, into 2 or 3 segment pieces, and each one of them can regenerate again. So if you find some of these, and you have a scalpel, go for it. It's pretty cool to see these animals regenerate. Another type of animal that you'll find, annelid that you'll find in freshwater around here is the hirudinids, which are the leeches. Here's one example that has these cool eye spots on the head. You'll recognize them because they have a posterior sucker so that they can attach to things. Pretty much none of them is going to be a problem for you, so you shouldn't be afraid of them. Most of them feed on, other worms or on snails and things. There are some that will feed on vertebrates, but anyway, they're not to be feared. Enchytraeids is a group called the potworms. They're kind of clear and white. You see them both in freshwater as well as in marine, terrestrial sediments. The natids, that includes things like tubifex, where you see a whole mass of them here. Bloodworms, sludge worms, the water nymph worms. This is a picture from our lab, so I love these. These are so beautiful. They're in this group. Super common. You'll definitely find them if you go out looking. And then a beautiful one that is the smallest. So we've kind of gone from the biggest to the smallest. These are just a couple of millimeters long. They're called oil drop worms, and they have these beautiful colored oil droplets embedded within their skin. So absolutely, completely recognizable. If you see these, there's no ambiguity about what this is. They also move around by using cilia with their heads. So they kind of move their cilia, and that kind of moves them around. Very, very cool and quite common. All right, and then in terms of the marine and brackish realm, some common ones are spionids. They're called palpworms because they have these palps that they use for feeding and then also for gas exchange. So these are quite common. I've collected these a number of times in the Chesapeake Bay. Capitellids, this one's a little bit sad looking, but very, very elongated, but otherwise don't have much in terms of additional structures that are coming out of them. Amphorettids, these are called spaghetti mouth worms. And I kid you not, this one has those palps, but if you saw this one alive, at one point, all these tentacles come out of its mouth and then they bring it back in. So they have these feeding tentacles, but they store them in their mouth. So when you find these you can find at Cirque also, so just near Annapolis, quite abundant, and they are just a riot to watch. They're fairly small. They're like a centimeter or so. And then something that's also called a spaghetti, but not spaghetti mouth, but actual spaghetti worms. You can imagine why. These tentacles are always out, so they don't come into the mouth. But there's this huge mass of tentacles. The animal lives in the mud, and then it puts out these tentacles all around it and picks up little bits of organic material, and then it brings it to its mouth. So a huge mass of tentacles. You're probably looking at a terabellid. Serpulids, feather duster worms. This one is also at Cirque. This picture even is from Cirque. They live in tubes, and they have these beautiful frilly appendages, tentacles that come out that they filter feed with. So they're trapping tiny, tiny little things out of the water. And Nereids, ragworms, which have these cool eyes. And I think this is my last one for this before I close out with my research. Glycerids, called bloodworms, beautiful animals, but not one that you want to get too close to, because out of their mouth, when they want to feed, comes this big balloony thing. It comes popping out of their mouth. And at the tip of that are these spines with some, even some venom. So these can give people a bite. I think this is the first one that, I would be careful. Well, I mean, it's not terrible, but they are carnivorous, and they have those structures to be able to subdue their prey. All right, I think I'll just jump over and just say they're super abundant. When we've done some, we did a survey across the Potomac River Basin, hundreds of individuals per liter of sediment at the peak, especially fall and spring. That's the time when there's the most. And also just done in a Virginia stream back in the 90s, a very substantial part of the biomass of the myofauna, so the small animals that live within sediments. And so the reason why, well, I'll just say that freshwater annelids still remain pretty poorly studied, and that's because they are, you can get down to genus for many of them fairly easily, but they're hard to identify because they don't have all these hard body parts that are often really useful for identifying to species. I've highlighted some of the identification resources, but there's not like the great online keys or other things. They're fairly fragile. They're difficult to sample intact. When you're swirling and decanting, it's not uncommon to break them. And it's a fairly small community that works on them. So I've shown you a lot of these beautiful pictures, but a lot of the time you're finding these, especially if you're preserving them in ethanol, you see these little bits of things. It's not inspiring. So I encourage you to fall in love with annelids by looking at them live and doing what you can to identify them based on live specimens. They're just so much more beautiful that way. And I will say that I love Maryland Biodiversity Project and iNaturalist, but the resources for annelids there, because of all those reasons, there's only very little there, and poorly represented even in these databases, and even frequently misidentified because that is a challenging group. But given that, I still hope that you'll head out and try to identify some, find and appreciate some of these annelids. So I'm going to close out and just telling you a little bit about some of my research on the evolution, development, and diversity of annelids. And I think I might just jump over one of them, just given the time, so that we have time for questions. We're really interested in what we call post-embryonic bodybuilding. So animals that can build their bodies, not starting from an embryo, but by doing something at the adult phase, or at least beyond the embryo phase. So things like asexual reproduction, where animals tear themselves apart or make worm chains, or bud other individuals off of their bodies, that kind of bodybuilding. Another category of bodybuilding that we see in adults is regeneration. When you cut off the head, cut off the tail, you can regenerate. So really interested in those processes. And I think I'll just jump ahead and say, we did a big survey across of published literature of annelids to understand how how does regeneration map onto the annelid tree to understand where has this, how old is it within this group? Does it get lost? There are annelids that can replace their heads and tails. There's some that can only replace their tails but can't replace their heads. There's some that can't replace either end. So how does that, what's the evolutionary pattern? So we did a big collection of data from the literature and added to ourselves. And I think I'll just jump ahead to the key result here, which is that regeneration is present. We reconstructed as present in the ancestor of annelids. So we're talking probably 700 million years ago. And that what we see as the pattern is losses. So we reconstructed 18 losses of the ability to regenerate and 5 losses of the ability to regenerate a head and 18 losses of the ability to regenerate a tail. So what we see is a pattern, and this is the first study that looked at regeneration across a whole phylum. We see like the signal that we see, at least within this group, is losses. So regeneration has been lost repeatedly. There's no evidence of any gains of regeneration. And I didn't show you that, but fission, the ability to regenerate, the ability to reproduce by making these worm chains evolves in lineages that already have regeneration. And so that suggests a pattern where a lineage has to first evolve the ability to regenerate before it can then start using it as a form of reproduction. I think I'll just jump over this and just show you the last one. So this is a study that was done by my grad student, and this is not your average worm. This is a small naid, so these water nymph worms. But it's kind of unusual. It's got this big bulbous head, lots of muscle, this big expandable stomach. It makes worm chains. So this is a new individual. That's a new head there. There's a new head getting going there too. So they make these big worm chains, but the morphology is really unusual. And I'll show you a video of one of these, and it is carnivorous. So here is our Chaetogaster worm. This is a little bit of a feeder worm, and you're going to see it detects it, and it will strike and ingest it. So it's going to detect it, and it uses suction, and it just puts it right into its body. Here's another one here, where it's trying to ingest an even bigger worm, but that bigger worm just breaks itself. It's going to be fine because it's a great regenerator, but it's losing the entire middle of its body. But these animals are transparent, so you can look right into them and see that bit of annelid that's been ingested. It's going to swallow now and put it into that stomach. Gulp, right? So remarkable annelids, not your average annelid. So we were interested in this group and how it evolved. So there have been 3 main species that have been recognized in this group. This big carnivorous one that I just showed you, a small one that just seems to be an omnivore, and another one that lives on snails. And we were interested in finding out about the diversity. Although 3 species have been typically referred to, when we did molecular sequencing, we actually found that what we thought was 3 species is 24. So we have a huge amount of what's called cryptic diversity, where we can't see the difference easily, but there's very clearly many, many species represented here. So lots more species than we realized. We also then did some additional sequencing of the worm guts and actually the whole worm to see what are they feeding on. And you'll see here's that big one that I just showed you. It's got most of what it feeds on is animal material. Here's one of the ones that lives on the snails. It's feeding on its host, but the close relatives don't feed much on animal tissue, which is shown in red. But instead, some of the closest relatives of that big carnivorous one feed a lot on little protists, including ones that move around. And so at least the hypothesis that we have is, at least in this case, maybe the way these animals became these really unusual predators is they started feeding on small single-celled animals that were moving around, developed abilities to have that suction action, and then probably progressed to larger and larger prey. So a lot of fun thinking about that. I'll close out here thanking my lab. These are my 2 current grad students, but a lot of great students over the years, funding sources, and you all for your for listening and having me out.
Well, thank you so much. That was wonderful. If you can unshare, come back, and we'll put a spotlight on you for some Q&A. I'm sure that you have some new acolytes, Annelid acolytes from AA group here. I'm sure that I'm one of them. We also have a spotlight. Here we go, spotlight. We do some macroinvertebrate sampling over at Leakin Park with the community. So we've found some leeches out there too. So that's always exciting when we find that. If you would like to put a question in the chat box, you may, or you can raise your hand and I will call on you. Let me just go back here. Jackie asked earlier on, what is the difference between Chaetae and Parapodia?
Great question, because they're often in the same place. So the Parapodia are the fleshy bits of the body wall that extends out. So think of it like if you could pinch your skin and pull it out, that's the parapodium. And then coming out of that so that's still living tissue. Coming out of that are these bristles that are secreted that are more like our hair, except, it's like not living tissue, but it's made by the animal itself. So those are the bristles are the Chaetae, and then that kind of pinch of tissue, these little flaps of tissue are the Parapodia.
Okay. Richard says or questions, are there major differences between freshwater and saltwater annelids?
Yeah, another great question. Most marine annelids have those Parapodia and have those bristles. There's really one big invasion of freshwater and land within annelids, and that's the group that gave rise to the water nymph worms that we work on, some of those other close relatives, the leeches, and the earthworms. All of those don't have Parapodia. So there are some of the marine ones that don't have the Parapodia, but you tend to only find the Parapodia in the marine realm, the marine animals. And then the freshwater terrestrial ones never have those Parapodia. So it's one big group that invaded freshwater and then invaded land. And so they share a lot of features. They're the ones that do the pairing like an earthworm. They're called clitollates. So that's mostly what you see in freshwater.
All right. Now, Nala asked about earthworms, but you mentioned that that's not your thing. I want to say that Dr. Slavich is coming back. She just did one on isopods for us, and she's going to be doing a talk on earthworms. So come back for that in February. It'll be up there soon. So we'll skip that. All right, let's come down here. Everybody learn so much, learn so much, learn so much, learn so much. Timothy asks, how many species of freshwater annelids have been recorded in Maryland?
And I tried to get that number for you. I don't know. There may be databases where that is accessible, but I can't give you an answer for that. I can say worldwide, there's about 6,000 species of leeches, earthworms, and these freshwater worms. So that's the scale of it. If I had to guess, I'm guessing if it's probably on the scale of a couple of hundred. That would be my guess.
But Timothy, I'm sure we don't know how many are out there. So there's probably a lot still to be discovered. So keep looking and go out there and look.
That's right. I mean, our one foray into that one carnivorous group, it's like we're off by a factor of 8, right? That's a huge undescribed diversity.
And speaking of that, well, in the data that you showed of the gut contents of the young bass, we had Matt Ogburn do a presentation for us on the eDNA study that you showed. So like,
Oh, good.
familiar.
That's great.
Study. And so you mentioned the DNA. Are you relying more on that for ID and than previously than looking at the physical characteristics? Are you just jumping to DNA now?
We did, in our study, we did, my student did take pictures of, because these animals, these water nymph worms are transparent mostly, you can in a live worm see into their guts. So many of them just have mush and you can't tell what it is, but every once in a while you could see in those carnivorous ones, oh, there's a water flea. Okay, oh, there's a nematode worm. Oh, there's like, you can see things, clearly a piece of algae. So we did do some analysis of what we could see in there. But the molecular approach is just so much more powerful, given that you miss so much when you only look for those visual signs. I still think it's really important to have the two so that you can have that strong confirmation where you can get it. But there's just so much more resolution with the molecular.
Yeah, it's amazing what they're doing with the eDNA, with this, seeing what went through the water. And Bethany asks, you mentioned this before, but maybe you can just reiterate, what makes annelids different from other worms?
Yeah, that's important. So for with those couple of weird exceptions that I showed you, annelids are the segmented ones. So you're not going to find another worm out there that has those segments. I would say if you're actually out collecting, there's 2 other worms that you're likely to encounter. Actually, there's 3 other things that you're likely to encounter. So I'll answer the question maybe like, what is most commonly misidentified as an annelid that's not an annelid? One of them are nematodes. Those are the roundworms. They're super characteristic. They just bend back and forth because they only have long muscles. They don't have circular muscles. So if you see something that's just like bending back and forth and back and forth and back and forth, that's a nematode. You won't see the segments. So that's one worm to say, okay, that's not an annelid. Flatworms. So some of them can be kind of rounded. They'll usually be kind of cruising around and are ciliated. So you, again, won't see the segments, but they'll just kind of be cruising around. So those are the flatworms, things like planarians. And then the third thing that you might confuse for an annelid are midge larvae. So that's an arthropod that has a larval form. It has a flying adult, but the larval form is aquatic. And that one will have segments because arthropods are also segmented. The way to tell that that's not an annelid is the outer surface just looks like they're kind of more encased. Annelids tend to be very soft. So if you can imagine, if this is your worm, and if you even just kind of touch something, it would just kind of like deform. That's going to be an annelid. You don't have to do that to check, but you'll see that outer body wall is very compressible. So that tends to be the annelids. And so those midge larvae, the other way you can tell that that's not an annelid, it's got like a head capsule. If you see a little kind of hardened, almost like a helmet on that thing that looks like a worm, it's probably the arthropod.
All right. Would you be able to put your email in? People wanted if you wanted to ask you questions after, if you could just stick that in the chat box.
Absolutely.
And Valerie asks, were you and your lab the first people to describe that, I don't know how to say this, the Ketogasther species, or were you aware of the existence prior to the analysis?
So the genus, it has been known for a long time because the worms have been recognized for a long time. But we're the first ones to recognize that there are 8 times as many species. So we were the first. There's been a second study that was just done for Chetagaster in Switzerland. And so they are kind of building on of what we found as well. But those, and you know, we just define them based on molecular grounds. So they're literally called like species 1, species 2, species 3. We haven't done all the big morphological description, in part because, you know, that's not our expertise, but also we don't have very much to go on. So in terms of morphology.
Nala asks, any information about transitions to parasitic lifestyles and back again? Has that happened a lot of times in different groups of annelids?
A great question. So just like with regeneration, that seems to have been there before, and the pattern that we see is one of losses. With parasitism, I don't know if the only big group of parasites are the leeches. And actually most leeches are just carnivorous, they're predators, but some of them become parasites. I don't know of any that have reverted back to like a non-parasitic form. So that might be mostly a one-way street. If I zoom back out to animals in general, parasites often fairly quickly become specialized to their environment, which is another body, the body of another animal. And so they often develop adaptations that are pretty specific. So it's a bit of a one-way street. It's not to say, I mean, I'm sure there's been some reversions, I'm not aware of them, but primarily things become parasitic, they become quite specialized on that, and then they don't come back to a free living form. Yeah, I love broad patterns.
Asks, how is the carnivorous annelid that you showed able to have such a strong suction? And I wanted to ask about that as well, because I thought, and I could think wrong, that sharks were the first animals to have like a sucking action in terms of evolution. So that was very interesting.
Yeah, and these are fairly recent, right? So one of the reasons that we like the Chetagaster worms is that it's a very recent origin. So it's within the genus that just 2 lineages become carnivorous. What's cool about that is you can see the close relatives and see, you know, like we did for the gut study, see what, you know, what are some of those closest relatives looking like? What are they eating? How do they look? So what we see in the closest relatives is fairly weak suction. They do seem to kind of poke at things, but without this strong suction. What we see in the morphology of that big Chetagaster species, that is the active predator, like an ambush predator, is that they have that big bulbous head, and it's, even though it's transparent, it's just packed with muscle, and they just expand it very quickly. So it's basically just forming that suction. You know, they move slowly, they detect something, they get pretty close, and then they expand very fast. That just pulls the stuff in. And if on that picture that I showed you, you could probably see that there's like these constrictions. They have very, very strong sphincters, like the mouth can just like shut, and at the, so that they keep that first, whatever they get into their first part of their head, that they've squeezed here and they've squeezed here so that the animal is trapped. Every once in a while they think, you know what, this thing is like fighting too hard, I'm just going to let it back out, and you'll see stuff come back out. But then it's kind of assessing, do I swallow? And then if it decides, yes, I'm going to, it's not too big, it's not too spiny, then it opens this one, pushes it down into that next part, and then at that point it can't come back up. You can watch it within about a minute, the material, like the tissue, turns white. So I'm assuming that there's like a strong, pH change, probably enzymes. It's a very rapid process after that. But in terms of actually ingesting, it's a suction. They are the coolest. And they're super common. You can definitely find them. For those guys, you find them in, not in streams so much, but in ponds. So those you would collect like the vegetation and look for these, they're kind of about the size of a big rice grain.
All right. And Timothy and Fatima are interested, are polychaetes restricted to salt or brackish water, or do freshwater species exist?
Great question. They are almost entirely marine and brackish. There are, I think worldwide, something like 8 species that are entering actually freshwater from that polychaetes that are freshwater. So almost, and I've never seen them. I don't even know exactly where they are. There might be one in North America, but so primarily, mostly, if you're in the marine realm, they're polychaetes that have typically the parapodia, although not always. If you're in the brackish water, you have some polychaetes and some what are called oligochaetes, this freshwater group. And then if you're in freshwater, there's almost no polychaetes. It's primarily that one group that has the earthworms and the freshwater groups.
All right, and Noelia is, what are some popular spots? People want to go out there and find these. Now we're getting into winter, and you said spring and fall were the best. So when would be the best time to start getting out there, or can we still find them right now, or do we have to wait? And where will we go?
Yeah, you would probably still find them now. They'll just be more abundant starting in kind of March or so. But you would definitely still find some in most places. So places that I like to collect in like local ponds, local creeks. I mean, really anywhere that has, if you have, like I go to a friend's house that has like a farm pond, those are great. Lake Artemisia, you can pull things there. Paint Branch, we've collected a lot there. It's all about just accessing. You don't have to go into any, I mean, if you have a boat, you can try to pull things off the deeper water, but just wading in or just from the edge is sufficient to collect that sediment.
And you definitely want to get the sediment in there, is that right?
Yep, sediment or some of that vegetation. Yeah, and if you go out and don't find something, if you have a body of water near you and you don't find anything, I would say, but that's the main place that's good for you to try to sample. I would say wait till March or April and you'll probably see things getting going then. A number of them, like the water nymph worms, are asexually reproducing, and so they make they have these kind of population explosions in the kind of March, April, May into early June.
You mentioned 700 million years. Is that when these annelids came onto the scene?
So the Cambrian is when we first start seeing major animal fossils. That's about 550 million years ago. But based on molecular data, there's ways to try to calibrate how long it's been since lineages diverged. And those estimates are more like in the 700 to 800 million years ago. So for most of the animal phyla, so most of the animal groups diverged probably somewhere between 500 million and a billion. So, 800 million is probably pretty safe as a number. That's the one I keep in my head. Well before the Cambrian. And there are beautiful fossils, Cambrian fossils of polychaetes, gorgeous with big chaetae. So that if you're interested in the kind of early animal evolution, if you just Google, polychaete Cambrian, you'll see some of these beautiful forms. So we've got some very nice forms, but there's not much before the Cambrian for really any animal group. For a couple there are, but mostly not. But we do, the fossil record starts around then for annelids.
Yeah, and I'll mention for Museum Store Sunday in our big fossil marketplace, we do have some fossilized worm tubes that you can come and get, big ones and small ones and all kinds of fun things. So
oh, how cool.
Yeah. Mike asked, do you see pollution effects, changes in species presence maybe?
Yeah, that's a good question. So we've mostly, I've been a lab-based lab, so we've mostly done experimental work in the lab, but over the last decade or so, I'm like, gosh darn it, I'm a biologist. I'm going to just find reasons to get out there more. So that's where we've started doing more of the collections. That's partly how the Ketogaster project got going. And so I had an undergrad who worked, who did a modest study here just right off campus in Guilford Woods, and just looked kind of where there's no more canopy because it's right next to it's basically like just a channel of water that's running between two strips of road, and then just not even like a kilometer upstream, it's still natural, which is in Guilford Woods. And so we looked across a number of months for what we saw in the main group that we work on, these water nymph worms. Very substantial difference. So although what a lot of people carry in their heads, especially if they're used to thinking about macroinvertebrates, is, oh, well, we've got these species, these groups that are arthropods that are indicative of really pristine water. And if we find those, that's a fairly healthy stream. The other end of that kind of spectrum is, oh, well, if you find a lot of worms, that's really degraded habitat. So, I take issue with that, because what's happening is that the local annelid community has been replaced primarily by things like tubifex, things that are very tolerant. But if they weren't there, there would be a different annelid community. And so we looked just along that gradient of like urbanized versus natural, and looked just specifically at the water nymph worms. And there are a couple of species that have very kind of large abundances only in the forested, natural, sloped area, and literally just, right next door, but where it's been canalized a bit, the canopy is gone, so it's now high. There's probably changes in terms of the groundwater, the substrate's changed. You don't see almost any of them. So we definitely are seeing that there are indicators of healthy streams in the annelids, even though kind of like what a lot of people carry in their heads is that if you see the annelids, that's a problem. So it's just certain species that come in.
Thank you for saying that, because you do carry in that when you do the macroinvertebrate studies and you're looking for water quality and things, and say worms are one thing, but that's fascinating and something that we need to pay more attention to. All right, any other questions this evening? As people are so excited to go out there and search for new things and make those data sets on iNaturalist and Maryland Biodiversity Project more robust.
That's right. I cruise around. I hope to see you on iNaturalist. If you post something that's a water nymph worm, I'm gonna come and cruise for that. I love doing that. That's my spirit, my fun Friday evening.
There we go. Okay, we'll know where to find you on iNaturalist. Well, thank you so much, Dr. Bailey, for coming and sharing your knowledge and expertise and passion with us. You do have some annelid acolytes here, and we're going to go out there and be searching for these wonderful creatures. And everybody, please come and visit us for Museum Store Sunday. This is the big marketplace. Come out there and get all your fossils, your minerals, all kinds of fun things that you can't get anywhere else for gifts, and learn about the petroglyphs of the Susquehanna from Paul Nevin. And in the meantime, stay well, stay curious, and stay outside. And we'll see you next week to learn about the Movement of Life Project. We'll study migration. Take care, everybody.